We are starting here resuming the topic of eye, and this time we are going to pick up here another section of the eyeball, that is the inner coat of the eyeball, inner tunica. Inner tunica means it is the nervous coat, or you can simply call it the retina. Now, as far as the retina is concerned, you have to have a little review about the other two coats which you have discussed in the previous class, the outer fibrous and the middle or intermediate vascular pigmented coat. And we have already seen here that if you discuss here in the vascular pigmented coat, I say in the fibrous coat, we told you previously that you have divided the eyeball into a certain portions, you can say here. That means in fibrous coat 5/6 posteriorly it was white of the eyeball, that is called the sclera, and here it was the anterior 1/6 that is called the cornea, that is a transparent. And you have already noted here that this layer which I am actually drawing, this is the outer fibrous coat in which I am actually drawing right now the sclera. That means this is 5/6 here, and after this 5/6, this part which I am drawing right now, this is the cornea. And we told you already that we divided the eyeball into 6 parts here through this type of a lining here. So, anterior 1/6, you see this is the anterior 1/6, and these all are you see remaining all will be posterior 5/6. And in this area especially, I told you that when you are actually starting here, limbus was there, this is the corneoscleral junction, and here immediately after you had here one thing that is called the scleral canal or canal of Schilling, and then there was a scleral spur also. This is your anterior pole and this is your posterior pole, and this line which we drew here in between like this is called the equator, means dividing them into the half, anterior half, posterior half. And similarly, we told you that not only this, but at the same time nasal to or medial to this posterior pole, this sclera was weak, because from here left your optic nerve fibers. This is called the lamina cribrosa. This portion means the portion of the sclera where the optic nerve fibers they left here. This was something about the fibrous coat. Now, as far the next vascular pigmented coat is concerned, not just this equatorial plane or line was important, there was an informal line also which we draw here, which will help us a lot here functionally. This line will in fact divide the eyeball into anterior, you can say up to here, anterior 2/6, and the remaining from here onward it will be posterior 4/6, or you can say anterior 1/3 and it will be posterior 2/3. This anterior 2/3 as anterior 1/3, this part of the eye, it is called anterior segment of the eye also. And this posterior 2/3 of the eye, that is called a posterior segment of the eye. Not only the anterior posterior segments, the anterior segment of the eye, if you are actually watching in this area, anterior segment of the eye which you are seeing, this is the part of the eye that is exposed, that is the visible part. You see, you can see it in the mirror from outside here, you see, in which there was total 100% cornea, you can see, and out of 5/6 of the sclera, 1/6 of the sclera you can see also, means total 2/6 you are watching. remaining 6 you cannot. And if this is the portion of your eyeball that is actually covered with the conjunctiva as far as your sclera is concerned, because sclera is avascular. So, that 1/6 part of the sclera will be getting its nutrition from the conjunctival blood vessels, because you see that this will be covered with the conjunctiva, not the cornea. Conjunctiva will be there also, but it is fusing and it will become a part of its epithelium. So, it becomes continuous with its epithelium at the limbus region, at the limbus you can say, at the corneoscleral junction. This was something in the fibrous coat we have seen here, but not only this line which is actually dividing them into the anterior posterior segments. In this line usually you see this line actually is passing across the lens also. So, lens is lying usually in this region. And is held from sides by the zonural fibers. So, in the anterior posterior segments, it is basically the lens and the zonular fibers which are dividing, which are actually be dividing them, and on the sides I will tell you as far as this is concerned. And whatever is anterior segment, I will tell you later. This anterior segment which is present here, which I will tell you later what happens in that, but then will come the next layer that is called the fibrous, is called the vascular pigmented coat, and out of this vascular pigmented coat, this 4/6 means which is invisible part of sclera, under that the vascular pigmented coat is present with the name of choroid. This will also be perforated obviously by the optic nerve fibers, and this choroid continues here, where up to the line which is drawing them, we say up to the line which is actually dividing them into which is dividing them into 4/6 and 1/6 region, you can say. This visible or invisible part means this will be actually up to invisible as a part I am actually drawing here. Now, this is up to visible part, it is actually present, this is called the choroid, 4/6. This is highly vascular and pigmented. But we have to complete all full circumference, 360 degree means we have to complete all 6 parts here. This is only 4/6. But in the remaining 1/6, the remaining 1/6 after the choroid finishes here, underlying the visible sclera obviously, because next 1/6 is this, there is ciliary body whose muscles are taking origin from the spur, scleral spur. This is called the ciliary body. Under sclera, visible sclera 1/6 under that, but the rest of it will be where you see the sclera ciliary body finishes. The next part of vascular pigmented coat comes here, that is called iris, which has the opening in the middle, that is called the pupil. So, this whole blue color is actually making a total all 6 parts here. Out of this 4 parts are the choroid, means under the invisible as a unexposed sclera, ciliary body under this visible sclera, and iris under the cornea, where you have seen in the center the pupil also, which has divided here, which has divided basically this one, means it has divided the lens, which I am actually showing you here, which is dividing here in such a way that which is dividing this and this anterior segment into 2 chambers. So, iris has divided them anterior chamber and posterior chamber, and the lens is actually the limit of your posterior chamber. So, I am making the lens here, these are lens fibers. And this is the lens, and you know that because the lens is actually suspended here by zonular fibers from the ciliary body, from ciliary processes. and this will give you idea that this small area will be your posterior chamber and this big area will be your anterior chamber, separated by the iris, but is communicated through the pupil. This is the pupil you are watching. This is something, the second layer, that is called the vascular pigmented coat. But now we are actually putting here, which is actually our today's topic, this is called the inner nervous coat. That inner nervous coat, its their name is the innermost tunica, which said this is called the retina. So you will actually see this now as the retina, and for this retina you have to pick up here one thing, that this retina basically, you see, this retina basically is a okay. So if you are taking this retina, it is the gift from, you say, it is gift from the brain to the eyeball. So this is the gift from the brain. Brain actually gives here an extension in form of a stalk, which bulges at its terminal end. This is called the vesicle. This is called the vesicle, and you call it now optic vesicle. And this is called optic stalk. At that optic vesicle, which is like a full sphere, you see that full circle here, and this will form features retina, and this optic stalk will form features optic nerve. Means this nerve which I am actually showing you here, this is optic stalk, which is connected here to the brain. So if this is optic stalk here, so where is our retina? Retina will be present here inside whatever space we have actually shown you. Inside here. Inside here in which way? You can say okay, in like a circle. But the circle will occupy how much? You see, in the total 6 parts of the inside of your eyeball, only 4/6 now can be occupied. The reason is that because the lens and the zonular fibers will not allow you to actually come into the anterior segment. Now this time the retina will be present only in the posterior segment, that is only 4/6, though in form of a circle, okay, no problem. So you can say in form of a circle like this. This is retina, which is forming the inner coat here, and now optic nerve is actually leaving, optic nerve fibers after penetrating your choroid and then the sclera. This should be called as the retina. That means if the retina is there, so that part of the retina which is in fact in the touch with the lens, means this portion which is in touch with the lens, this is called its nervous part, neural part, and the remaining which is in touch with the choroid, this one, this is called its pigmented part. Non-neural, you can say. Up to here you see this, it's a non-neural part. This is called its non-neural part. The green color which I am showing you, non-neural part. That means non-neural part, it is overlaid by the by the choroid, in touch with the choroid obviously. That means in touch with the choroid basically is the epithelial part, epithelial layer. Actually this epithelial layer which is green color, which we have shown you here, green color, which is epithelial layer, this is retina's first layer. Will this red be the second layer? Actually it will form the next 9 layers. It is not just second layer, it will be forming next 9 layers. So next 9 layers will be just behind the behind your lens? No, because this retina which is actually in form of layers, this is the neuronal, I say neural layer, it is actually here, and this is the epithelial, we call them retinal pigment epithelium, but they will not remain like this. In fact they will collapse in such a way that initially they were like this, but later on they will be like this now. This will be like this. It will be like this. Means if you are actually looking at this, so this will give you idea, that means this will be our, as usual, our epithelial layer, that is called the pigment retinal epithelium. And the one which has actually gone behind here to make itself in touch with the epithelial layer, this one, this will form 9 more layers. You can say 10th was already there, 9th, then came 8th, then 7th. Likewise, you will reach up to the 2nd. Why 2nd? Because the first is already there. I mean, the 2nd will now touch the first already present, and first was this epithelial is called the retinal pigmented epithelium, RPE, and 2nd will now touch, and 2nd belongs to the neural layer, and 2nd actually which is belonging to the neural layer, that is comprised of rods and cones. These are the photoreceptors. Means photoreceptors themselves have gone and invaginated themselves into the retinal pigmented epithelium, and the space which is present here, you see, coming to this diagram, the space which is present here will get narrowed down and will become like a slit. This is called the intraretinal space. Here intraretinal space is full, you see that big balloon is there inside. The entire space should be called as the intraretinal space, and here when you are actually bringing it, intraretinal space will be smaller and smaller and it will become like a slit here, and a slit means it is intraretinal space has actually been slit. And once the receptors outer segments, I say the segments means the outer segments of the rods and cones will invaginate into the retinal epithelial, the retinal pigmented epithelium. Intraretinal space will be no longer. So they will become now continuous 10 layers from outer first layer, then the innermost 10th layer. In between we have 8 more. Okay, the intraretinal space has been obliterated here, but if I am actually telling you like this, now watch here in this. Okay, now this is space, this layer, that is called the neuronal layer, and already I told you that it has gone behind, but gone behind at this area where it has bent here, means non epithelial layer, that is actually called the non neural layer, photoinsensitive layer, is becoming continuous with the photosensitive layer. That area is called the ora serrata, means this point here. And here it is. This is called the ora serrata. Serrations are there, we see interdigitations. This is called the ora serrata. But still, I am showing you here intraretinal space. But I tell you that when more layers, neuronal layers, nerve layers will be actually neuron layers will be developing here. This intraretinal space will be no longer. We have 10 layers of the retina which will develop here, and now there is no longer intraretinal space. Now, as far as this intraretinal space was concerned, we told you that here, but when this intraretinal space is gone, initially you see that your retina was just like an optic vesicle, but now it will become, now previous optic vesicle just like O, but now it has become double C now, you see, because it has collapsed here. It has been deflated, you can say. And after deflation, when it become double C, you see a big space is created here anterior to the retina. Big space has been created anterior to the retina, and that big space in fact will be filled by here another transparent gel like substance, that is called the vitreous. So there will be vitreous now. This is a vitreous which we are filling here. which is not only supporting your lens from behind, but in fact it is supporting your retina also. So much so that these two layers, as long as your vitreous is intact, these two layers which have coalesced to become now two layers of the retina, the neural and non-neural at ora serrata, these two layers will never be separated as long as your vitreous body is intact, and even the spherical shape of the lens will be maintained by this also. Okay, this is your vitreous. And this is, so a whole posterior segment has been covered, actually be filled by the vitreous here. It has been filled by the vitreous, but at the same time you see, that means you can say now posterior segment of your eyeball has the fluid that is called the vitreous humor, and the anterior segment of the eyeball has the fluid that is called the aqueous humor, and they are giving actually a globular shape to your eyeball here. In this, especially when you are actually discussing here, we told you that in this discussion especially, this is the area which you are going to pick up. This is called the retina today. The other topics you have seen previously, 10 layers of retina with no intraretinal space. Now embryologically it was there, but nowadays now it is not there, and here the optic nerve fibers are leaving here. Eyeball develops like this here. Now if for example you are bringing, now come back here and see some more. Now after this brief introduction, we are going to pick up here the inner nervous tunica, the retina, which you know that is occupying the posterior segment of the eyeball. You can say the posterior 4/6 of the eyeball. You see, this is here up to ora serrata, you can say, means we are watching this yellow color up to posteriorly, means from ora serrata onward here, composed of 10 layers in such a way that the outer layer or the outer surface of this retina is attached here to the choroid, firmly attached, but the inner surface of the retina is towards the vitreous, means it is sandwiched as such between the vitreous and the choroid, and this will be actually here the inner and the outer surfaces, this way. And the area, you see that there is a point where abruptly the light sensitive area ends and is becoming continuous with the light insensitive area, means the pigmented area or the pigmented layer, that is called the ora serrata. And at the ora serrata it will be overlaid here by the ciliary body, because from here will start obviously your next 1/6, fifth 1/6, where retina itself will not be present. Retina itself is not present, but the representation is there. The reason is that because this pigmented epithelium, which is actually the first layer in case of retina, will continue here in the ciliary body also. It will continue in the ciliary body, not only just continuous in the ciliary body, but also with the iris also, pigmented part of the iris, pigmented layer of iris as well as ciliary body. Though in ciliary body and iris is continuous, it continues the neural part of your retina also, but their function is but they are non-functional, you see. And this is the portion of this colored portion, I said this pigmented portion of the iris and ciliary body is present here also, but this is continuous with the pigmented part of the retina, which is that is why this is actually called the pars iridica retini, and that is called the pars ciliaris retini, but the behind which is actually present is called the pars optica retini. So this is something you are actually watching in the brief nutshell. You see that of the retina here, but watch a little more. Ora serrata, already you have seen this. Ora serrata is a junction between the retina, the light insensitive and the light sensitive area. non-photosensitive and the photosensitive. And the non-photosensitive, you know, that is already the simple non-photosensitive area, which is made up of retinal epithelial, retinal pigmented epithelium, but the complex photosensitive region of the retina is the neural layer, you can say, and both undergo transition from each other at ora serrata, and ora serrata is the level like this here. Ora serrata is the level that over, after that it will obviously be the end of the choroid and beginning of the ciliary body will be there, and after that already you know that is the iris. So, this ora serrata, and this ora serrata actually demarcates, you can say, two segments also, posterior segment filled with the vitreous, anterior segment filled with the aqueous. This is ora serrata here. See again here, this area that passes through the lens. Now, if for example, at the entrance you see that, for example, you have seen here on the back pole or the posterior pole, little to the medially, not in the center of the posterior pole, medial to the posterior pole, there is a weak area over the sclera, weak part of the sclera, where there is a exit of the optic nerve fibers here. But that exit will not be just present, you see that at say over the sclera, but similarly exit will be from your choroid also, and similarly such exit will be at a point in retina also, and that exit point, or you can say entrance of the optic nerve, is not entrance or exit, means it will be exit obviously, because signals are going away from eyeball. That 1.5 mm circular pale area where there is exit of your optic nerve fibers, this is called the optic disc. And this optic disc means right from optic disc onward, the optic nerve fibers will take the name of optic nerve, and you are watching this here. So, optic nerve, so we call it the optic nerve. You see, this is this pale area. From here, all the optic nerve fibers leaving the retina from any spot should in fact will leave this portion, just like the hilum of any organ, you can say. And it is not just this optic nerve fibers are actually leaving here. So, you can say optic disc, it is forming the head of the optic nerve, because from here onward you see optic nerve will be actually leaving, the fibers are leaving here. So, all the fibers will leave from here, and this is called the optic nerve fibers. And if for example, you are watching in this also, so you see right in the center of your optic disc, there is a central artery of retina visible also. So, there is central artery of retina visible as well, and this is the central artery of retina is entering here. This is the branch of very important branch of the ophthalmic artery, and ophthalmic artery, you know, that is the branch of the internal carotid. And this ophthalmic artery enters here through the optic canal, from where the optic nerve is already entering, and before entering into the the orbit, after it has entered into the orbit, is giving many branches, including this important branch, that is called the central artery of retina, that will enter also in the center of the optic nerve fibers, and when it is entering here into the eyeball, it splits up into the upper branch and the lower branch. The upper will actually divide itself, you see that upper will divide into the nasal branch and temporal branch, nasal branch and temporal branch. This is central artery of retina, and then it will split up into many small branches, then the arterioles, then the capillaries, and all this one, all these four are the end arteries. They are not anastomosed with each other. So, if any embolus, if for example traps here into any of these arteries, coming from the internal carotid, you see, coming from coming from your left atrium, as a left ventricle, as a result of, you see, this embolus that will detach from here, comes into your ascending aorta, then arch of aorta, can enter here into one of the common carotids, then common carotids will go more into the internal carotid, from internal carotid it can more chances to go actually in the brain, but it can slip and can here come into the ophthalmic artery. And in the ophthalmic artery, then you see that ophthalmic artery will be there, and it will be not just ophthalmic artery, but at the same time ophthalmic artery will give one of the branches, that is called the retinal artery. It's called the central artery of retina, and it can trap itself into any one branches, any of the branches of the the central artery of retina, upper division or lower division. And this upper, it should be either nasal or temporal, and once it is actually blocks it, that part of, you see that retina will become insensitive. It will become say necrosis, the ischemia will take place and necrosis will occur. Blindness, sudden blindness will be there, because it is an end artery, no other artery will anastomose and can compensate. And these are the arteries, you see that these are the arteries, arterioles and other things you can see through ophthalmoscope. These are probably the only arterioles which you can see in intact healthy body, and to look at them you can see their composition, you can see their status, and through the status you can actually diagnose a lot of diseases. And this ophthalmoscope, and in this ophthalmoscope you are actually seeing this field, whatever field in this whole, you see this in a circle you are watching, this is through the ophthalmoscope you are doing it. So optic disc which you are seeing here, and I told you that when the optic disc is there, means say if this is here, you see inside is retina is there, and through this here you are actually seeing. So optic disc, you know that it will be medial, means at this area it is optic disc, and inside you know that intraocular pressure is there. So intraocular pressure will be in the optic disc, because outside the area is weaker, perforated. So this optic disc will be usually depressed, you say it will be concave, it will be excavated to a variable degree, producing something that is called the physiological cup. There is a physiological cupping of the optic disc. And in the optic disc, because all the optic fibers are actually leaving the retina, and then the choroid and everything, and then the sclera, means this area, the door, it will be just like a wall, everywhere there is a bricks, but when in the wall there is a door, there are no bricks. So here that piece here, part of the retina from where all these fibers are actually coming out here, that will be kind of a door. So no bricks will be there, means here you will have nothing actually, no rods and cones and anything will be there. You will call that spot will be insensitive spot, and that spot will be insensitive to light. That's why it is called the physiological blind spot. So optic disc is basically physiological blind spot. If there is any other blind spot in a part of retina other than this physiological blind spot, that will be called pathological blind spot. You call them scotoma. This will happen when any part of the retina will have any problem. So in this area you see that when through ophthalmoscope you are putting the light here with the magnifying glass and light, you are actually seeing the innermost layer or the innermost aspect of your retina through the iris as through the pupil here. So you can put the light here, and we are putting the light, you can imagine that if you are putting the light and we see these structures, so if you are put the light here, people will constrict. That's why to in fact minimize the pupillary constriction, or you can stop or prevent the pupillary constriction, you will put the ocular drops here, the drops which will block the light reflex. Light reflex means the muscle that is called the constrictor pupillae muscle, which you are putting light and it will undergo constriction to narrow down the diameter of the pupil. You will actually paralyze them by putting the drops like parasympatholytic drugs. You are actually putting like atropine, hometropine, etc. Then you will do this here, and what you will actually see, you are seeing the disc and the whole surrounding this, whole the structure of the retina surrounding this. So this is optic disc, central artery retina, splitting branches here, and you can see not only this, but at the same time you can say lateral to this optic disc, you will see here 5 to 6 centimeter pale area, you will see this here also, slightly avascular area you will actually see. This is called the macula. Because it is pale, it is yellow, that is why I call them lutea. Luteal means yellow. This is called the macula lutea, and in the center of the macula lutea you will see here a depression, means if this is here, then this is a depression here. This is called the pit. We call the pit is called as the fovea, because it is in the center, that is why I call them fovea centralis. Macula lutea, and in the center of the macula lutea is fovea centralis here. So you will not just see optic disc, but you are seeing these structures surrounding that also in this whole circle through the lens of the ophthalmoscope, and that thing is called fundus. That is why this ophthalmoscopy is other name is the fundoscopy also. And through fundoscopy you can see all these arterioles. Central artery of retina is coming here, so dot will be there, and the central vein should be also there, but at the same time central venules and central artery, the retinal venules and retinal arterioles you can see also through ophthalmoscope. But this is a fundoscopy you are actually seeing here with the magnifying lens and the light here. But I told you that to check the to prevent the pupillary constriction and to keep it enlarged, medicated eye drops of hometropine or atropine, these are the parasympathetic antagonists you will actually instill in the conjunctival sac few minutes before due to the examination. Macular lutea, already I told you 5 to 6 millimeter lateral to the optic disc, obviously, because optic disc is more medially, because here will be the eyeball here and this is the pole here. So, this will be more medially here, the optic here is the optic disc here, but this will be just behind the upper just behind the anterior pole here is the posterior pole area. This will be means if it is medial this one, so this will be little lateral here. So, you see this lateral. How you will differentiate? Differentiation will be big because in the center there is central artery of retina which is splitting into the branches also. This is more medially and this is laterally. See here again, it's a green color. Whole this green color is a macula, but this depressed area in the macula is a fovea. That area is the fovea, and this you can see here just opposite the anterior pole in the center. The optic disc is lateral, or say medial to this. At the same time, you can see here this optic nerve. We have told you that in the retina there are 10 layers, and out of these 10 layers, the first layer which was already present here, that was called the non-neural layer, that is called the insensitive, light insensitive layer. This is called a retinal pigmented epithelial layer, which is single cuboidal cells here, single layer of cuboidal cells. Or you get short cuboidal, you see, a short columnar. This is here, and then remaining will be this neural layer, and that neural layer will be comprised of 9 layers. And it is comprised of the 9 layers, and out of these 9 layers you can say the second layer will be this was the first. Second layer, it will be the outer segments of your rods and cones, and then these rods and cones, when this outer segment is coming, then when the inner comes here, you will see here a line or a membrane which will hold all them together. You can say holding them by neck, you can say. So, that should be stabilized here. This is the third layer, that is called the outer limiting layer. And after that, you will see that the nuclear zone of each of your rods and cones will come. This will form your fourth layer, that is called the outer nuclear layer. And then their synaptic terminals will come, and finally will be signals will be sent here to the dendrites of a cell group, that is called these are the cells, very important here. These are called the bipolar cells. You call them bipolar cells, but obviously where the anastomosis, I say where the synapses will take place between the synaptic terminals of the rods and cones with the dendrites of your bipolar cells here, that area will in fact be connected with each other by means of this another group of cells here, which will make here a kind of a network in a zone of synapses between the synaptic terminals of the rods and cones with the dendrites of the bipolar cells here. So, this kind of a network will be created, a plexus will be created. This is called the fifth layer, and you call it now outer plexiform layer, which in fact is formed here and operated by surely by the this yellow color cells. We call them horizontal cells. So, these are the horizontal cells. We saw here rods plus cones, we have cells also. And at the same time, when this fifth layer is gone, now this is the game of now bipolar cells, and bipolar cells will should have here their cell bodies also, and the cell bodies of bipolar cells, they are lying in this sixth layer. This sixth layer should be called as the inner nuclear layer, because this fourth was called the outer nuclear and this is called inner nuclear layer. and that inner nuclear layer, in fact, from here their axons are now running, and they will now join the dendrites of, or they make the synapses with the dendrites of yet another cell group. Now this is called here the cell group, this is called the ganglion cells here. But the ganglion cells, dendrites, and the axons of the bipolar cells surely making here a synapse, and here making synapse of another bipolar ganglion, here also making a synapse of the bipolar ganglion. So, if this is here, so they themselves are connected with each other by means of another cell group, like a horizontal, here smaller cells are there. So, another network is created here. These smaller cells are called amacrine cells, and these are called amacrine cells. But I told you that this network which has been created by the amacrine cells to connect the synapses here, this is called the plexiform layer, and you will call this plexiform layer as the 7th layer. This is called inner plexiform layer, and then surely your ganglion cells are coming. This is the 8th layer. So, what will happen with the axons of your ganglion cells? You see the ganglion cells axons are now coming, and all of these layers ganglion cells will actually coming here, their axons are coming, and everybody is proceeding towards that gate. This also, this also, this also, all these cells here. At which area? At the optic disc area. All will come here, like from this side also, everywhere. This is optic disc. So, 9th layer will be optic nerve fibers layer. You see the optic is called the nerve fiber layer, NFL. So, that all these nerve fibers in fact will go here into the optic disc. But why you are expecting that will go here toward the door here? Why not they will actually go forward here and will come will penetrate the vitreous? Because there is a membrane. This will be your 10th layer. This is called inner limiting membrane. Just like there was outer limiting membrane which was holding the rods and cones from their necks to stabilize their and to keep themselves in line here. Similarly, optic nerve fibers will not be let enter here, enter into the vitreous here. So, this is what we call here the 10th layer of retina will be there. We will be actually discussing on them. In this, the receptors which you have seen here, with the receptors which you have seen here. So, you can watch here, you see this receptors. Consider the receptors just like your receptors elsewhere in the body, like pain receptors, here rods and cones. Application of stimulus, here application of stimulus, here application of stimulus by what? By you can say pinprick, here application of stimulus is by the light, light is the stimulus. So, here is a, for example, pinprick is there. Pinprick, it is a stimulus, light is the stimulus. Both have been actually applied on the respective specific receptors here, and the receptors function is to transduce these stimuli into an impulses. This is called transduction. First was the application of the stimulus, and then will the transduction, and transduction will mean the impulse has been generated, and impulse should now travel along the wire. Wire here will be now your peripheral axons of some neuron, whose cell body you know that is lying outside the central nervous system in form of a ganglion that is called the dorsal root ganglion, so that its central axon should enter into the spinal cord. I say in any central nervous system portion here, this is called the first order neuron. Same is the case here in the retina. Here it was receptor, here is the receptor, but the receptor here in case of pain it is called the free nerve ending. Here you call them either rods or the cones. These are the rods, these are the cones, whatever they are, they will be actually going here. So, they, if you are putting the light here, application of the light, transduction of the light stimuli into the impulses, and the impulses should now be traveling and be handed over to the next order. I say handed over to the neuron here, to the neuron means to the dendrites of the first order neuron, and dendrites of the first order neurons are, these are the first order neurons, we call them bipolar neurons. So, they are dendrites, so relate to them. And these dendrites should be present where? Outside the central nervous system. Okay, difference here is there, they are lying outside the central nervous system, this is true, but they are, you see that they are lying outside the central nervous system, they are present in the retina. Retina is outside the central nervous system, retina anatomically is outside central nervous system, this is true. This is lying inside the eyeball, not inside the brain, but consider retina being a part of the central nervous system. This is a gift of the brain, you can say, extension of the brain as such. Through the optic vesicle was what? The continuation of the brain. Here this was this was actually made up of the neural crest cells, but they are not. here in form of the ganglion, but it is in form of a cell body separately here, and it is a cell body separated. So, dendrites are receiving them and cell bodies are there, axons are now leaving. Axons are leaving, where the axons of this dorsal root ganglion enter? Central nervous system. Here not central nervous system. Why? Because they are already in the central nervous system. Retina is a part of central nervous system, and they will be actually leaving. Here they are entering into the central nervous system. In the central nervous system is awaiting their second order neuron. Their dendrites will receive them. Here second order neurons also are present within the central nervous system, means within the retina. So their dendrites are receiving them. This is your second order neuron. This was your first order neuron. Same. Now this time second order neuron will do what in this in your spinal cord, for example. The second order neuron, it will now give rise to their axons, which will run up here in form of a tracts. This will be your parent tract. Say for example, spinothalamic tract. So here the axons will do what? Okay, here axons also will make the bundles, but unfortunately you will not call them tracts, though they are actually coming from second order neuron, and they are actually also forming the bundles, and they are inside the central nervous system also, because retina is the central nervous system part, though it has actually been implanted here inside the eyeball, but it is actually here. So from our point of view, we can actually call this as the tract here, but we will not call it a tract, unfortunately. And it will leaving here the optic disc, and after the optic disc here you call them the optic nerve fibers, and when it is leaving now the optic disc here, you will call them optic nerve even, but still you will not call it the optic tract, though they are the second order neuron. So much so that when you are if you looking at them, right at the point you see inside, when they were running in the retina, forming there you say 9th layer, which has been limited here by the inner limiting layer, that is the 10th layer, so that they should be easily and safely be guided into the lamina cerebrosa, and also from inside you will see through the optic disc, then through the choroid, then through the lamina cerebrosa. But once they actually leave now the retina at the optic disc here, so that will be the head of optic nerve. Now their name will become now the optic nerve fibers. You call them the optic nerve now, still you do not call them optic tract, though it is the second order neuron fibers, and second order neuron fibers always are called tracts. Actually nerves which have been formed here, they will never be formed by the second order neurons. Nerves are always formed by this first order neurons, but this will be formed by second order neurons. Actually from practical point of view, this is tract, but still we call them the nerve. If you will call them this tract, it will, if for example had we called them tract, there would not have been any, you see, there would not have been any you say optic nerve present in your central nervous system. In your nervous system there would not have been any optic nerve here, because if it is right from here it is optic tract, so where the optic nerve will come, but still we call them optic nerve here. And I have told you that this is called the optic nerve here, but you see that if one eyeball is this, another eyeball is this, medially is arising your optic nerves, and immediately they enter here into the optic canal, and once they enter into the optic canal, you see half of the fibers are nasal, they come from nasal retina, and half of the fibers they are coming from temporal retina. So those fibers which come from nasal retina, immediately after leaving the optic canal, they are crossing, but those fibers which are coming from the temporal retina, they do not, and they will follow the crossed fibers. So they are the uncrossed and crossed, both are running together. And now after this rearrangement, at the time of half fibers crossing, this is called optic chiasma. After rearrangement, now you can call it at last optic tract, though it is not third order neuron, it is a second order neuron, and second order neuron, now you can call it the tract. Now we are equal. Here is a tract, right, since beginning. Here it was a nerve in the beginning, and later on it will become tract, only change of names after the chiasma. And this optic tract will actually be leaving, and it will be synapsing in what? All the sensory modalities will synapse and report to the thalamus, here also to the thalamus, and here the specific nucleus of the thalamus will be there, whose name is lateral geniculate body. And here this paint carrying tract will also go into the thalamus, where there is specific place of the thalamus will be present here. There it will be called as the different thing is called the ventral posterolateral nucleus of the thalamus. But now this is the responsibility of the thalamus. Now from here the third order neuron should in fact will send their axons, because this nucleus itself was the third order neuron. First order neuron was this dorsal root ganglion, second order neuron was the nucleus present in the spinal cord, and third order neuron will be now in the thalamus. From thalamus they will be sent here to the specific cerebral cortical area, and here it was also sent to the specific cerebral cortical area from thalamus onward here, from geniculate body to the cortex, in a specific area in the occipital zone of your cerebral cortex, and here various details of this image will be actually decoded or will be recognized. So, this is something that you are watching. Already I told you that central artery of retina, you are seeing this temporal nasal branches, these are the end arteries we already discussed here. Central artery of retina, when you are actually discussing in this area, for example, when you are discussing this area, up to here you say wherever your axons are present, I say wherever your cell bodies are present, not the outer segment or the nuclei of receptors or whatever more areas you are actually watching, no, not them, but where you see the cell bodies of the neurons, that inner area will be supplied here by the central artery of retina. All these areas will be supplied here through diffusion by the choriocapillaries from the choroid, because overlying here will be the choroid and rich plexus will be related here. That is why its name is choroid plexus name. You know that choroid is a vascular pigmented coat, and they are getting its blood supply from the choriocapillaries. We will tell you this, but here this is called this central artery of retina, and in this inner layer, total retina already you know there is a 10 layers, and out of this one is called the pigmented here, and remaining 9 layers, you can imagine remaining 9 layers will have almost 9 different type of cells, not like each cell is lying in each layer. Always the students are fond of actually dividing the things like here 9 layers, 9 cells, so each layer will be taking one cell group. No, it is not like that. So, you see here, if for example I am telling you in this, the two real game players are obviously this one, number 1 bipolar cells, first order neuron, and number 2 the ganglion cells. Surely they are the 2 cells, but you know that when the plexuses were formed between the your rods and cones and the bipolar cells, you required here the third group of cells. This is called the horizontal cells. This is third. When you were actually seeing here your bipolar cells were actually synapsing with your second order neuron, first order neuron is synapsing with second order neuron. Here you see lateral connections between both of them. You see here another group of cells, you call them amacrine cells. This will be your fourth cells. At the same time, we do not have just this, but you will see here in between in these areas small other cells also, glial cells, you call them astrocytes. So, you have got fifth also, not just astrocytes, but you will be actually looking at, we will tell you that there are very big cells in the middle, especially in the region of bipolars. Their nucleus is there and they have got a short processes and they have got long processes. These are very important group of cells here. These are also glial cells, we call them Muller glia, we call them Muller cells here. This will be your sixth type of cells. And at the same time rods and cones you can consider my cells also. You see 6, so 7th will be rods, 8th will be cones, and the 9th will be the small cells which actually have come from the blood. These are called microglia. So, you see 9th type of cells are there. But where these cells here, not the rods and cones, these two, but where the other cells are there, 7 type of these cells are lying here, central artery of retina will supply them. So, central artery of retina is very important from this point of view. So, you can say outer 2/3, outer 1/3 supplied by choriocapillaries, inner 2/3 are there supplied here, by where the neurons are there, they will be supplied by this here. And as far the central artery of retina is concerned, we have told you that, and we will tell you later also how this blood retinal barrier will be considered here, because we have got two blood retinal barriers also, inner and outer. We will discuss that in retina. Now, this was the central artery of retina, and central artery of retina at the time of development, actually it was running through the vitreous and reached here up to the posterior pole, and the posterior pole means posterior capsule of the lens, because the capsule of the lens and the epithelium, because the avascular should be getting its nutrition from aqueous humor. That is why the blood has been actually made to disappear from here. Just like cornea also needs the aqueous humor, because aqueous humor is not yet been produced here in the early embryonic development here. So, temporarily this artery will supply the lens also. But later on, when the aqueous humor will start, you will see that this artery actually will obliterate. Arteries, this distal portion will obliterate right from here. You see that right from this pole, means this is optic disc, from optic disc up to the posterior capsule, at the center, little nasal to the center, and this will be obliterated here. So, inside you will have actually a transparent canal. This canal is called the hyaloid canal. The hyaloid canal running through the vitreous from the optic nerve disc, from the optic disc up to the lens here. And a spot which is actually present here, this is a small circular opacity on the posterior capsule of the lens, and that posterior capsule of the lens, this small opacity, circular opacity, is called the Mittendorf dot. This is called the Mittendorf dot, and this canal, which is empty canal, you see it is kind of transparent canal, that is called the hyaloid canal. Actually its name is a Clockett's canal also. This is a hyaloid here. Actually central whatever you have seen here is green color, this is a hyaloid canal now, no longer an artery, but this initial portion, very important artery that persists, that will be called the central artery of retina. Actually initially its name is the hyaloid artery, and hyaloid artery means proximal part of the intact hyaloid artery's name will become now central artery of retina, and the rest of the portion will be like a canal, that is called hyaloid canal. So, see, this is transparent hyaloid canal, and we are watching here little to the nasal side here. Vitreous, we have already told you that is a colorless gelatinous substance that fills the space between the lens and the retina, because here is the lens and here is the retina. So, this actually fills this gap here, or whatever gap is there, this has been filled. And you can this is supporting the lens from behind. So, it is a spherical shape of the lens is maintained by this, and at the same time posteriorly it is actually pressurizing the retina, so that the two layers of retina should be in a position with each other. When there is a vitreous degeneration, so two retinal layers will not be able to touch each other, so they will try to separate from each other. This is called retinal detachment. And this is you see, you cannot actually take this whole ball or body outside here, or this vitreous body outside. It is in contact, you see, it is in touch with certain parts of the capsule. At the ora serrata it is in touch, or the optic canal, as optic disc, it is in touch. You see, it is firmly adherent, you cannot actually detach it. Not only this, but you see it is having it is in touch obviously with but it is not I can say the retina is not adherently attached with this, because there is a membrane, this is called vitreous membrane, which separates it as such physically from the overlying eyeball structures. But once you remove the vitreous, for example, after detaching all these attachment points here, it will not grow back by regeneration. It is a mass of protein made during our embryologic development, that we get once for all. This can have some say replacement of certain nutrients and others, but this will not be replaced as such. You get it once during embryological development. Now, this was something in general about the retina. Now, you are entering into the real world of retina, and you will actually watch each of these layers in bit of the detail, so that even in physiology, histology, all these things should be actually solved in this platform today, and it should be once and for all, at least your second year students are concerned. These are the capillaries, choriocapillaries. These are the rods and cones, and the rods and cones, they have their outer segment here, which is embedded here, so that invaginating inside the epithelial cells, pigmented cells. So, we have like this also, but at the same time you are actually seeing here they have their own cell bodies also, rods and cone cell bodies, and then there is a, you say, constricted portion which is forming synapse. All these rods and cones, by the short processes of this Muller cells, are held together so that they should be they should be aligned here. not scattered here and there. Their tips are already being trapped here in the epithelial cells, and their necks, you can say their neck has been tightly held together, so that they should always be in line with each other, you can say, in a disciplined way. This membrane which has been actually formed here, this is the layer number 3 actually of your retina, that is forming outer limiting membrane, OLM we call it. And similarly, when I will tell you here, when all retina will finish here, all 9 layers of the retina will finish, means one layer already there, 8 more retinal layers will finish, then the long processes will actually form this horizontal membrane here. This membrane is called the inner limiting membrane. This is your layer number 10. So these Muller cells are actually the glial cells which are stabilizing you also, and other neuronal structures, they will also be stabilized. And not only this, but I will tell you, they are in fact maintaining the inner blood retinal barrier also to some extent. So these retinal structures means they, in fact, these are the structures which maintain the structural and functional stability, number 1. Also inner nuclear layer, they have got, I have already told you that they are in fact having the long processes forming the outer inner membrane, and they have got short processes which are forming the outer membrane, long processes forming the inner membrane. These are called the limiting membrane. See here, limiting membrane, outer, and this is inner. Inner is the last layer of retina, and they have a lot of functions. We told you that stability also, removal of the debris which is will in fact present here and there, in fact is giving whatever extra neurotransmitters in fact will be released within the neural environment, it has been taken away by this. They give electrical insulation to the receptors and other structures which will be present there, mechanical support to the neural retina. So all this is the function of the Muller cells, very important group of cells. You see the largest cells being here like this here. See this cytoplasmic membrane, see this nucleus is present there. You can see this also, nucleus present there, their short processes giving them the membranes. Stability is a very important pillar, like you see. Now come back here and see the layer number 2. This layer number 2 is the photoreceptor layer, PRAL you can call it simply, short form being PRLA. Structure of the rods and cones we are discussing, but watch here, this is the epithelial, retinal pigmented epithelium. But later on you are watching here the rods and cones here. Rods and cones lying next means you can say retinal epithelium, retinal pigmented epithelium. Next layer is called the rods and cones. Outer segments of the rods and cones, not full rods and cones, it's outer segments only. Watch them. Now see the structure of the rods and cones here. Good structure. You see this is here. These are the rods which will look like see, because their outer segments look like rods. That's why we call them rods. And when the outer segments will look like cones, you can see here cone, you see this is like rod, look like cone. Because of this their names are rods and cones. As far the inner segment is concerned, now you see this is the outer segment. Here is the outer segment, and if you are looking at the inner segments, inner segment is this full, which is the 4 parts. Out of these 4 parts, this portion I said largest one, this is called the ellipsoid part in the rod, as well as ellipsoid part. You see ellipsoid part in the cones also. This contains a lot of mitochondria. The next portion, which is more narrower here, in case of a rod it is more longer, but in case of the cones they are constricted here, smaller. This is called the myoid part. This contains rough endoplasmic reticulum. This is more larger, longer here, it is more shorter here, this one. This is called a myoid part here. Understand, this is called the myoid part. This is will be actually the myoid part, which contains rough endoplasmic reticulum. After that, it will come the next thing, that is called the nuclear zone. Now this is the nucleus, and when the nucleus will come, we told you that at this level where the myoid part is there, at this level will be your outer limiting membrane, which has been formed by the short processes of the molar cells here. So, first layer, you know that it is the first layer was the pigmented cells, this was the first layer. The second layer was in fact the rods and cones, their outer segment and their you say their ellipsoid part as well. But the third will be the inner I say outer limiting membrane, so that all rods and cones should actually be standing here in line, should be stabilized. This is our third layer. This is called outer limiting membrane, which is in fact formed by the short processes of the molar cells, and then will come now your nuclei. This is your fourth layer now. So, that means if there are here, you see that if there are here tight junctions, so the plasma environment in fact will be not allowed to enter here in the rods and cones. At the same time, from the inner cells here, this environment should not also be coming here towards your rods and cones, because this has been limited here by the outer limiting membrane. So, that means this outer blood retinal barrier in fact from outside is prevented by the tight junctions between the RPE, but from inside it has been prevented here by the outer limiting membrane, which you know that is being provided by the molar glia. This is our layer number 3, and here is the layer number 4. I told you that the rods and cones nuclei themselves. This is called now nuclear layer. You call them outer nuclear layer. Outer nuclear layer means these are the nuclei of the rods and cones. And then the fourth part, I say fourth part of the inner segment is their terminals. These are called synaptic terminals. Shape is different. These are called the synaptic terminals. In such a way, the synaptic terminals of the rods, they are called as spherules, and the synaptic terminals of the cones are called the pedicles, because they are feet like, they are broader as compared to this. This is the difference. You see, these are certain differences between rods and cones structure. I am actually telling you in detail. The reason is that because they are asked in the histology, they are asked in the physiology, they are asked at multiple you see at different sites, you see that, so that we should finish them here. Now, see, this is the outer segment here. In the outer segment, you see these are called discs. They are suspended within the cytoplasm, but you see the discs here, they are not on one side, they are not having actually the membrane here, so they are not within it, so they are formed like here. So, they are like this, but here it is not like this. Here their discs are inside here, and these discs are containing the visual pigment. See the rods and cones, and we have already told you that pigmented layer epithelium is layer number 1, rods and cones number 2, outer segments obviously. Then is this outer limiting membrane, which is coming from the short processes of your molar cells. This is number 3, and then surely is coming now the rods and cones nuclei here. This is a outer nuclear layer, this is layer number 4, and then you see that their terminals are coming. If these terminals are coming, now this is the time their terminals will hand over their signals towards the first order neurons. These are called the bipolar cells, which we have actually seen previously also. They are first order neurons equivalent to their dorsal root ganglia in any sensation, normally if you see. Now, as far the rods and cones are concerned, we have already told you, the rods basically they do not register color, but they are sensitive to dim light here. This is called scotopic vision. But as far your cones are concerned, they can register color, so they are called a photopic vision, photopic, scotopic. And if you are actually looking at this, we have already told you that there was a macula lutea, and inside the macula lutea center it was the fovea centralis. Fovea centralis contains only cones. This is the rodless area. This contains only cones here, because this is basically the rodless area, and you say it is the cones here. So, it is only the cones present here and the huge concentration. And if you are actually looking at this, that this is now the rodless area where you have seen here the cones only, but when you are going to the periphery, now the rods and cones are almost equal in number. so much so that in the periphery almost there are no cones at all, it is the cone free area. In the cones, you see in the fovea centralis, which is almost isolated, totally cones, here no rods at all. But these rods basically are not rod like, they are actually, you can say in the fovea the cones are in fact, the cones are there, but these cones are not like cones as such regular cones, there they are like the rods, even you can say, that is why they are called the roddy cones as well. But periphery, I told you that cones and rods are almost equal, more and more and more, and till we reach the periphery almost cones are totally absent here. See the structure differences more. And as far as your rods are concerned, cones are concerned, there are a lot of differences inside them. You see, rods are too many in number, you can say 120 millions total rods as compared to your say 6 millions only. So, you can say 120 by 160, you can say 20, means it's almost ratio is 1:20, but in the fovea centralis is ratio, you know that 100% cones, rodless area. Outer segments of the rods, already I have told you that it is rod like, that is why I call the rods, and outer segments of the cones, it is cone like, that is why they are called the cones here. So, pigment is visual pigment, in the rods it is the pigment that is called rhodopsin, but here it is called the iodopsin. Rods, they will in fact register black and white vision, but the cones will have the color vision. See again, see if you want to remember, it is called the cones. You see that is why the black and white vision, when you are going into the dark room here, you can see the things, their margins, everything, but you will not see the colors. That is why in dark light you see that it is the work of the rods here, but the cones are for color vision. The rods, you know, that is lying more in the periphery, not in the center, that is why the peripheral vision is better detected here, means peripheral vision. So, they are good for peripheral vision. Peripheral means the size of the pupil should be dilated, so that the periphery of the retina should be utilized here. So, better vision when the pupil is dilated, because they are lying more in the fovea centralis or in the area around the fovea centralis more, that is why the central vision is actually good for the cones here. So, this is a better vision will be for the cones, it is better vision when the pupil is constricted, so that the light should actually pass through the central portion of the retina. They are more sensitive, they are less sensitive. Why? The reason is this, because the ratio of the rods is 1:3 or even more, 1:3, 4, 5, 6, 7 it can be, but here is 1:1. What is this ratio? Ratio will be that this is the, for example, this is your rod. If this is your rod, now this is the terminal. This rod's terminal in fact will synapse with the dendrites of the bipolar cells, and if you look at the cones, they are also doing the same. This is synaptic terminal, and here is again the bipolar cells. Here in case of the cones, here in case of cones, in case of rod, the difference is here is a 1:1 ratio, means one bipolar cells only can be stimulated by one cone, and I tell you that the bipolar cells cannot receive rod and cone both together, either they can receive cone or they can receive rod. So, that means they are receiving just the one cell. No, here if for example, if the cone is there, it is almost 1:1, but when it is a rod here, so why just one? Another rod can be received by the dendrites, yet another rod also, yet another, for 5, 6, 7 here can be received by this. Means one bipolar cell which is receiving the rod, it will not receive just one, it will not 2, even 3, 4, 5, 6 can be received. So, that means stimulus which is coming from the cones, for example, if the stimulus is +1, means this bipolar cell will get actually the signal of +1 only. But here in this case +1, +1, +1, all they will summate here and will be +6 here. So, stimulus here to the bipolar cells which is receiving the rods, it is actually multiple, it can be summated here, it can be 5 +5 +6 +7. So, 6 to 7 times more than the even the rods here. That is why the signals of the rods in fact they are the light which in fact is perceived here by the transmitted here by the rods, it is much more, because the stimuli they are more intense stimuli which are in fact going towards the bipolar cells here, and here the stimuli are very miniature stimuli are there, because there is just one cell which is one cone which is actually doing this. And as for the rods and cones which have been stimulated, I told you that they are having the graded potential. Rods and cones, they communicate via graded potential, not the action potential. It is not all or none law, means it will be depending upon the intensity of the photons here, intensity of the quantity of the amount of the photons, depending upon the intensity of the light. More intensity, more potential, less intensity, less potential. Action potential will be either yes or no, it is all or nothing. Either will be none or it will be full, here according to the intensity. It is a graded potential, just like your even receptors, all the receptors have the graded potential. Rods are more sensitive, I told you that is because of one cone synapse is one bipolar, but many rods stimulate one bipolar. So, stimuli they are summated, hence rods are more sensitive. That is why through the rods you can see in the dim light also, because in the dim light, and that is why in the dim light you can't see the colors, because cones are not utilized, because they are less sensitive. Already light is less, how they will be sensitive. This was your second layer, and this now come to the third layer, that is outer limiting membrane. Already I have told you these are actually, this is short process of your Muller cells, in fact will form them. Short process of Muller cells, I have already told you that short process of Muller cells, they will actually be holding the you can say the constricted myoid part of your inner segment up to here. Come from where? They are coming from the Muller cells here. They go horizontally and will catch hold of them. This is your third layer. I have told you the second layer is the rods and cones. This is photoreceptor layer, and first layer already you note it is called the pigmented layer, and all of them they are getting their nutrition from the choriocapillaries. Some more also, even your nuclear layer, that is the nuclear layer, that means next layer will be what, the nuclei of the rods and cones. This is your layer number 4. You call them as the outer nuclear layer. This is ONL. You see, this all is outer nuclear. This is called the outer nuclear layer. Already I told you this is the nuclei of your rods and cones. A part of your inner segment is there. Then comes here a very important layer, that is called the plexiform layer, outer plexiform layer. What it is now, you see this. Now, for example, if you are actually discussing here, you see this now your rods and cones, and you know that rods and cones, if the rods have their spherules now coming here, these spherules are cones whose pedicles now they are coming, they finally will hand over their stimuli which they have already transduced into an impulses. They have to be now shifted here to the first order neurons dendrites, and these first order neuron dendrites are in fact the dendrites of these cells. You see the shape of these cells, because first order neuron, if they are special senses, they are first order neurons, if they are special senses, and if they are special senses, not just they are special senses, but if for example not just they are special senses, but they are having their special sensory nerve also. If these two criteria are fitting into them, their first order neurons must be bipolar. So, there are 4 sites where these two criteria fit. Number 1, smell. Smell has a special sense plus their first order plus their sensory nerve is a specific purely sensory nerve, olfactory nerve. That is why their first order neuron must be bipolar. Number 1 example. Second example is hearing. Hearing, it is a special sense and it has got a specific nerve, cochlear nerve. That is why their first order neuron is again bipolar. Cell body is present in the organ of the corti, it is bipolar. Then vestibular, again second part of your second component of your eighth nerve, vestibulocochlear, is the same. Cell body is lying within the bone here in the modulus. You see that the cell body is bipolar. Why? Because it is a special sense as well as it has got its own specific nerve, purely sensory nerve, bipolar. Come here in your vision, see bipolar is present here. Why? Because it is the vision and it has got its own specific purely sensory nerve, optic nerve, who does not do any other function except carrying the visual senses, visual stimuli. But you say that the fifth sense is the taste. Okay, taste is special sense, we agree, but it does not have a specific nerve. A specific purely sensory nerve is not present for the taste, That is why test first order neurons, they are not bipolar, they are just like general sensory first order neurons, they are also pseudo unipolar. Pseudo unipolar, already you know that it has one cell body, then it got one fiber which splits into two, like here. This is in all first order neurons of all general senses, plus now test sense is like this. So, there are 4 examples of the bipolar neurons, and it is very important example, you know, that is the this layer. And bipolar layer, already I told you that pigmented epithelium number 1, rods and cones number 2, outer limiting layer number 3, and we have told you that when you are actually entering now into this layer, we told you that this is called 4th will be outer nuclear layer. These are the nuclei of the rods and cones, and now their terminals are ending into the dendrites of the bipolar cells here. And bipolar, you know that this is the first order neuron, not lying in the ganglion, because the bipolar, wherever they are present, they are not in form of a ganglion, you say the sensory root ganglion, they are scattered in form of cells. Bipolars usually they do not form ganglion, they remain almost scattered here. At some area they can form, like for example in the organ of Corti, the ganglion of say semicircular ganglion of organ of Corti, we call it like there is in the ganglion, but usually they are scattered. And this is now the layer, and this is the outer nuclear layer, I have told you then this will be actually called the layer, usually is the bipolar neuron, okay. But you see the bipolar neurons, they have got their dendrites where the synaptic terminals of the rods and cones, they are ending. So, this zone is very important where both are actually meeting. Who is meeting? Terminal ends of your rods and cones with the dendrites of the bipolar cells, and where they are present here. So, multiple such connections you will see here, here, here and here. If you want to these connections, if you want to connect them together also in a lateral way, for this purpose to connect these units together, you need another type of a cell here. This will be horizontal cells. You watch this green color, horizontal cells. They will in fact get the connections from these synapses and will in fact connect them with the other synapses here. So, they will operate laterally in such a way that here a network will be created in a zone. Which zone? In a zone of the synapses between the synaptic terminal of your rods and cones with the dendrites of the bipolar or the first order neuron here. So, a plexiform layer has been created here, that is called the outer plexiform layer, and this will be your layer number 5. And here is called the layer number 5, and this plexiform layer which I am actually telling you, it is called the layer number 5. After that what will happen? You can follow the dendrites. Where you will come? You will come to the nuclei of these bipolar cells, and the nuclei of your bipolar cells. So, a layer has been created, a very important layer. This will be your inner nuclear layer, this is the 6th layer. So, inner nuclear layer is in fact the layer of the first order neuron bipolar cells here, and I have told you that in that zone was in fact lying the cell bodies of your molar cells also. You see big cells which are actually lying here. And after that what will happen? This is the 6th layer that you have actually seen here. So, you have seen the inner nuclear layer. I have told you that inner nuclear layer means the nuclei of the bipolar cells. So, location of inner nuclear layer, you know that between the nucleus is lying where? This nucleus is lying inside, you have got another cell, that is called the ganglion, and outside you know that is called the receptors. So, you can say they are lying in the middle because they are first order neuron. You know that always the first order neuron, in case of even your normal sensations, general sensations, receptors are lying in the periphery, first order neuron lying in the cell bodies in the dorsal root ganglion, inside the spinal cord was lying the nucleus. So, your first order neuron, which here is the bipolar, which is lying here the inner nuclear layer, will be lying between what? Between receptor and the second order neuron, and here second order neuron is this, which I will tell you is ganglion cells, and receptor is the rods and cones. So, bipolar cells are actually in between them. Signals from the receptors will be received by bipolar, and bipolar cells will actually hand over these signals here to these cells, these called the ganglion cells. And we have already told this bipolar cells will be 1:1 ratio for the cones and 1:6 ratio you can say for the rods, and they are handing over these signals where? So, these signals means their axons will be handing over there to the dendrites of these cells, which is the last cells, you know that here, which are closing here to the membrane, to the outer inner limiting membrane. So, they are actually there, dendrites are there, and these dendrites will receive the axons of the bipolars. Then another bipolar will be in fact putting its signals here to the dendrites of the another ganglion cell, then yet another to another, likewise here. So, this zone will be again important, synapse zone, you can say, zone of synapses. This time between what and what? This time between the dendrites of the bipolar and the axons of the bipolar and the dendrites of the ganglion. Previously it was you see that the dendrites of the bipolar and the synaptic terminals of the receptors here, and where there was the first type of synapse zone was there, this zone was connected with each other by means of these neurites of the horizontal cells to form here outer plexiform layer, and similarly here this time not horizontal but small cells will now come here. You see these small cells, you see this, these small cells in fact will connect these synapse zone with each other to form here another plexiform layer. This is called the inner plexiform layer, and this inner plexiform layer will be your seventh layer. We have told you that first is the pigment layer, second is your rods and cones layer, third is your outer limiting layer, fourth was the outer nuclear layer, fifth was outer plexiform layer, and sixth is your inner nuclear layer, this is bipolar nuclear layer, and then you are actually watching here their synapse time, and synapse time in fact will be amacrine cells, which will make the synapses connected with each other to form this here. So, they operate laterally like this, and another plexiform layer is constituted here. This is called the inner plexiform layer. These are amacrine cells. A means no, mac means big, these are amacrine, they are not big but the small cells. Amacrine is actually small cells, they have got short processes which in fact are doing like this. After that what will happen? After that obviously the dendrites which you have already made the synapses, they will connect what? They will now connect these cell bodies, okay, new layer, eighth layer, ganglion cells. This is a second order neuron, and such second order neurons normally lie where? Normally lie inside the central nervous system, and here they are lying in the periphery. No, they are lying also in the central nervous system, because retina is a part of central nervous system. And then eighth layer. So, what will happen? Now this is the turn of the axons of the ganglion cells here. Okay, come here, see, as far your cells are concerned, you see bipolar cells, already I told you that it is a graded potential just like receptors, and like most of the neurons, bipolar cells I have to communicate with via graded potential again, not action potential. Seventh layer I told you the inner plexiform layer. Who forms them? Amacrine cells. Amacrine cells, they have short process, short neurotic processes here, and they have inhibitory neurons, so that whatever signals are reaching here, and if the signals are actually more than enough, they should be inhibited, regulate them, and they work laterally. So, a plexiform layer will be created here. Then is the eighth layer I told you, this is called the ganglion cells, very important cells. You see this is the final cells, obviously they are in fact their collection should be called as the nucleus. In case of other sensations here, where the inside central nervous system second order neurons, they receive the signals here from outside here, and they are called the nuclei, and their axons are called the tracts. But here they are forming the eighth layer, okay, ganglion cells is the eighth layer, and from their axons bundles are actually leaving, and axons bundles which are leaving them, they will finally enter here into the optic disc here, and once they leave the optic disc, their name will become optic nerve. Actually from our side conventionally they should be called as the optic tract, because they are the bundles of the second order neuron, and second order neurons always form the tracts in sensory system. They are not forming the nerves at all. Nerves are only formed from the first order neuron, never from second order neuron, but here they are called the optic nerves here. Still you call them optic nerves once they will leave the optic disc here. So, the head of the optic disc, you see that optic disc constitutes or say represents the head of the optic nerve, and you know that inside these fibers. So, this ninth layer will be the nerve fiber layer, you can say, and nerve fiber means it is the nerve fiber layer, means it is the optic nerve fibers obviously, but these are called the axons of the ganglion cells here. So, ninth layer will be like this. And once they are remaining, you see, once they remain within your retina, they are unmyelinated. They will attain the myelination once they leave the optic disc. So, outside they will become myelinated here and myelinated. So, we are expecting that the outside myelinated, okay, no problem, they will be actually from the Schwann cells, and Schwann cells come from the neural killer cells. No, they are actually outside, they have got, because they are getting their myelination origin or myelination source from inside, and they are actually inside. What inside? This is in fact the optic nerve. Basically this is a tract, but we do not call them a tract. So, myelination around the tract is actually coming from where? Oligodendroglial cells. Actually you call it nerve, but consider its myelination is from the oligodendroglial cells. This is enough to actually remind you, to convince you that please call him a tract, not the nerve, but still you call it a nerve. And this is probably the only nerve which has the myelin sheath formed by oligodendroglial cells. And if the oligodendroglial cells in fact have the autoimmune problem related here to the multiple sclerosis, this is the only nerve, because it is fond of having actually its myelin sheath by the oligodendroglial cells, so it has to pay the price. So, multiple sclerosis, this is the only nerve, only part of the peripheral nervous system which is actually affected. No other peripheral nerve, in fact no other nerve will be affected except this. Reason is this, because it is having a myelin sheath coming from oligodendroglial cells. But after a short while you see that we will be in fact will be convinced that okay, this was not the nerve, it was actually the tract. So, when the nerve is in fact just leaving the retina at the optic disc, it will have its nasal and temporal fibers. Once we actually wait only that their nasal fibers, they will cross and go to the opposite side, and temporal will join the crossed fibers. Once they do this, still they are actually the axons of your ganglionic cells. They are second order here. Here they were second order, here they are second order, even here they are second order. But once they cross, actually we are waiting for the chiasma to form. Otherwise, when the chiasma has been formed here, now you will call this fibers, now we are convinced, now you call it the optic tract. Here we call the optic nerve. Actually right from beginning it was the tract, but this early portion its name is the nerve. Otherwise, if you will actually ask them it a tract right since beginning, probably we would not have the optic nerve in our syllabus. Probably the optic nerve would not have an existence at all if you call it the tract since beginning. Okay. So, if the ganglion cells, you see that what they are doing, they are actually getting the signals from bipolars and sending them here to the thalamus here, because tract finally will be going into the thalamus. Some detect the objects, especially some of these subsets are very important. They will be stimulated only when the objects are moving, and some will detect the objects which are stationary even. And some have the pigment which will in fact sense the light and will not send them to the thalamus for image or for object vision, for vision purpose, but for the non visual you can say things here. Like for example, many of them, some of them, they will in fact will leave them and will form the optic fibers here, optic nerve, and they will actually be going not to the thalamus but to the hypothalamus in a nucleus that is called the suprachiasmatic nucleus, which is lying in the hypothalamus just closer to the chiasma. This is called suprachiasmatic nucleus, and this is the nucleus which is actually controlling the hormone system according to the light, according to the day and night here rhythm here, that is called the circadian rhythm, and that pathway is called the retino-hypothalamic pathway. Some of the ganglion cells are like this also. And we told you that they transmit image forming and non-image forming visual information. This was non-visual forming, retino-hypothalamic, non-visual. Visual is not related to this, but non-image forming. Visual are there, but it's non-image forming. But others are image forming. Obviously, their main function is image forming. But some functions, because the current is there, so this current will be used for regulating the hormones in a circadian rhythm here. But mind it, it is always doing it through action potential, not through graded potential. They are all at one law, and another will be their action potential. Usually we know that action potential is actually the depolarization of this of the cell membrane, but here action potential is actually hyperpolarization. This is probably the only action potential which is created by hyperpolarization. And it is actually, so some of the subsets of ganglion cells sending the signals to the hypothalamus, and some of them can even send here to midbrain also, but most of them to the thalamus obviously. They have very long axons which you know that will leave, initially we call them optic nerve, later on optic chiasma even, but finally we are convinced they are tracks. So, we will actually call them the tracks later on, but after chiasma. Functions already you understand, small percentage of ganglion, they are not themselves, even some of them they are themselves photosensitive. Otherwise, they will give the signals to the thalamus, and from thalamus to the here you can go to the cerebral cortex, but your photosensitive things are actually the rods and cones. But if if all the rods and cones in fact they are damaged here, the light which is entering inside them directly can stimulate some of the reserve type of thing. You see all retinal detachment full total complete, still some vision will be there, because ganglionic cells they themselves are, some of them I say a very small percentage, not a kind of a vision, like shadows will be there even if all rods and cones have been damaged here. This is called the ganglion cells, which are in fact having a small percentage being contributing to the vision also, because being themselves they are even photosensitive. And after that nerve fiber layer, you know that these are the axons of your axons of the ganglion cells, which will be, and if the axons of the ganglion cells are now coming, we are afraid they will actually go into the vitreous, and I have already told you that as long as they are in the retina they are unmyelinated. Once they will actually leave the retina at the optic disc, they will become myelinated, and myelination will be not from Schwann cells but from oligodendroglial cells. And you know that when we are expecting that maybe we are afraid they will actually go into the vitreous, no, there will be now coming your 10th and the last layer, that is called the inner limiting layer, and this inner limiting layer has in fact been provided by again the Muller cells long processes here, so that all these fibers in fact will be going here towards the door that is called the optic disc. So, all they should actually go through that. So, inner limiting membrane I have already told you, it forms here, like it forms the barrier between the vitreous and the retina, because here is the vitreous. Now, as far as this is concerned, we have told you here there are tight junctions here, also your inner outer limiting membrane was there, OLM. So, this atmosphere was totally prevented from any assault from inside and even from outside here. Similarly, we have in fact, because all this actually is the inner 5-6 layers in fact having its blood supply coming from the retinal artery, central artery of retina. So, when the retinal artery is actually coming here, so you see that is entering here through the optic disc. I have told you that here these are the fibers which are actually leaving. So, this entering will be your optic central artery of retina, and splitting here in its inner layers into the arterioles and the capillaries, retinal capillaries here. And these retinal capillaries are important in such a way. So, you say sir, blood will enter now very easily outside here. So, your plasma environment will be in connection with your inner neural layers here. So, there is no blood retinal barrier here, there is blood retinal barrier, because these capillaries, these retinal capillaries which are there, they have got very tight junctions here, they are tightly packed here, number one. And outside you see another cell type which name is the astrocytes. That short processes in fact will bind the retinal capillaries, so that whatever, even if the gaps are there, they will be filled also. To some extent even your Muller cells will in fact offer this thing as well, offer this blood barrier as a blood retinal barrier. This is inner blood retinal barrier you call it, and this was outer blood retinal barrier. Understand? So, these are now the layers, you can say the layer number 1 epithelial layer, layer number 2 rods and cones, layer number 3 outer limiting, layer number 4 inner outer nuclear, layer number 5 you say outer plexiform, layer number 6 is the bipolars, layer number 7 inner plexiform, layer number 8 ganglion, layer number 9 optic nerves, layer number 10 inner limiting layer. You can see this also. 1, 2, I say 3 here, 4, plexiform layer 5, 6 is your bipolar, 7th is your inner plexiform layer, 8th is the ganglions, 9th will be your optic nerve fibers, 10th is now the limiting membrane. This you are seeing. That means when the light rays enter now into the retina, it will pass through the anterior chamber, through the pupil, through the crystalline lanes, through the vitreous, which is again you can say again it is transparent, then striking the 10th layer, then passes through your 9th, 8th, 7th, everything here until it will be absorbed totally by the epithelial layer, and whatever is remaining will be absorbed by the choroid. Whatever is reflecting back will strike now the outer segments with the visual purple will be actually bleached, and this will lead here to the graded potential, and that graded potential in fact will be transferred here to the bipolar cells, return back here to the ganglion, and now it is going through the optic nerve fibers, which will leave the optic disc to go to the thalamus here, and thalamus will send them here to the visual cortex, and here the perception will take place. Though the thalamus is also giving the visual perception, but that visual perception does not have the too much details now. See these layers from outside in, and here is your vitreous. So light enters from here. So this was your class regarding the layers.